Hello. Yes, Rameka, I
Your
can hear
voice
you.
is not coming.
Hello. Hello.
Hello. Hello. Thanks.
Can you hear me?
Hello.
Hello. Hello.
Ma'am, your voice. Hello. Yeah, now I'll be able to hear.
Yeah.
Yeah. Ma'am, I have some doubt in the previous questions, like in the calculations. Basically, I'll try to calculate, but I'm not able to solve the all the question, all some questions, not all, like little three four
Uh,
questions.
okay.
So the
You have
questions have.
received the mail regarding the calculator, right? They have sent the demo calculator by mail, I think.
Yes, ma'am. I'm just telling about, sir, ma'am,
Yes.
the calculator. No, yes, I'm talking about the calculator also next, because I'll send the mail to NPTEL team. In that calculator, ma'am, if we calculate some value, it's give the wrong answer.
Oh, okay. I haven't checked it, but
Actually, I'll
I can
compare
check.
it to the normal calculator which I have in my hand,
Hmm.
and another I'll calculate with that calculator. But the answer would be very different. So I'm just worried about if the similar questions has come, then it's quite difficult to me to solve the questions.
Okay. We'll check that. You can ask your doubt first.
Yeah, one second. I'll just. Ma'am, the first questions I have to be asked, it's come under third assignment,
Okay.
which yes, third assignment. The question is related to the half life of Eretine something. Like first assignment, question number 10.
Just a second. Third assignment, you said,
Ah,
right?
yes. Third assignment, question number 10.
Question number 10th.
Yes, ma'am.
In a study with a groundwater sample, that one only. Hello.
Yes, ma'am.
In a study with a groundwater sample, it was observed that tetrafluoroethylene is degraded. That one only.
No, ma'am, not that one. Like
Read the question.
the half-life of the A aharicide degradations follow the uh, first order kinetic. It's
Half
established
life of 2,4-D
the
is.
no, not 240, ma'am. The questions, I'll read it once again.
Uh,
The half-life of the degraded following the first order kinetic, it estimated to be the approximately 14 days. What fractions of the initials uh, Aragonine with B will remain after 90 days. So the formula we use, C is equal to CO e की power minus KT, half life is equal to 14 days, CO divided by 2 is equal to COE minus K power 14, K is equal to ln 2 by 14, and the answer is, when you solve ln 2 by 14, the answer is come as 0.049 days.
yes.
So
Days
that I don't.
inverse.
Yeah, ma'am, but I don't know how to get the because if you solve ln 2 by 14, it's not come for me 0.0449 days.
What? Just a second.
So that's why I feel here one calculation something. mistake, I believe.
What are you getting the value of ln2?
Ma'am, as per answer, because as per answer, the answer is 0.049 days.
Don't see the answer, you calculate yourself, use the calculator.
Yeah, one second, ma'am. Then I'll calculate now only.
Yeah.
Because when the time of I'll calculate 2 divided by 14 is equal to.
No, wait. You are doing it wrong. It is log of 2 divided by 14. So you don't have to divide 2 by 14 first. First, you have to take the log of 2 and then divide it by 14.
Ma'am, there is ln hai, not
Huh,
log.
that only. ln is natural log. So when you take the natural log value of 2, first you have to calculate the ln.
Or
Okay,
then I'll divide by
14.
14.
Yes, so ln2 would be 0.693 and then you divide 0.693 by 14.
Okay. Thank you,
Okay.
ma'am. I'm not very good in that field. That's why I'll ask very
No
basic
problem.
questions.
But then,
Yeah,
uh,
now I'll get answer.
so
Yeah.
whenever you have this, so whenever there's an equation, you should see where the brackets are. So the uh, any equation, if you are solving, first you should solve whatever is there inside the brackets, and then you try to solve the things that are outside the bracket. There is a rule called Bodmas rule. If you remember, you must have studied in lower classes.
Yeah.
So brackets are solved first, and then you take the division value, and then multiplication value, then addition value, and at the end subtraction values. Okay.
Okay, ma'am. Second question. One second, ma'am. Second questions is mine. Uh, ma'am, like one there is a questions of question second.
Hello.
Yes, ma'am.
Yeah.
Hello, ma'am.
Yeah. Yeah, which question?
Ma'am, question second. In that question second of the assignment, the radioactivity of nuclide P32 has half-life of 14.3 days. Initially, P32 will remain after 84 days. Find the ratio uh, activity of the first order reactions.
Yes.
Ma'am, is there how is it? Is there no where I'm facing problem? Is there all solutions? I fully understand, but one part I'm not understand. There is a mentions e to the power minus 0.0485.
Yes.
That I'm not understand how they get like
That is the k value, right? So L. So here also same thing. So
like
half
how it
life, half life
half life is equal to
is
ma'am
given,
0.
right?
Yes, half life.
So half life is given, so you can calculate the value of k ln2 divided by half life, similar to the previous
Half-life is
question.
not given. Yeah, half-life is given 14 and K value is 0.469. That how it's come? It's a fixed value.
ln2 is the yeah, so that is the formula, right? So t half is
Okay.
equal to uh, ln2 divided by k, or k is equal to natural log of 2 divided by t half. So ln is basically natural log. So you have two types of logarithmic values, you can say. So if you have log with the base 10, it is written as log. If you have log with the base e, in that case it is written as ln. Okay, small l and small n. And in that case, if you have any ln value, that's how you are getting this. If you see in the formula, it is written ln in the bracket c0 divided by c is equal to kt, right? So that means C0 divided by C will be equal to e to the power of
KT.
kt, because the ln that is given, that ln is basically natural log that has the base e.
Yeah.
Understood?
Ma'am, like, yes, that I understand. Second point is I want to be asked, like, how to solve e की power minus 4.074. That e value is equal to 2.72.
So in the calculator, you will have a
Yes,
button saying e to the power of x.
e to the power of x.
Do you have it in that calculator? Let me also check.
No, ma'am, I'm using normal, like computer calculator. Is there is alone mentioned and x की power y1 is there given.
E to the power x is not mentioned. Check the NPTEL calculator, because that is what you are going to use, right?
One second.
So I am sharing my screen, so it would be easier. Okay, so this is the NPTEL calculator. Here you can see e to the power of x,
Yes, ma'am.
right? So whatever value you want to calculate, so let's say it is minus 0.0485 into 84. Okay, so first you have to calculate that. So minus, or first you write 0.0485. Okay, if this is the value, and I'll click this plus slash minus. If you click this, you can convert any value from plus to minus. Okay, so if I click it again, it will become positive. If I click it again, it will become negative. This is fine. I hope you can see the cursor, right, on the screen. Yeah, so this is now negative of 0.0485. Now let's say you have to take the e to the power of this. You want to calculate e to the power of this. So just click on e to the power of x, and you will get, see, power of e. Here you can see power of e in the bracket minus 0.0485. So you will get the answer as 0.9526. Okay,
I get it. Thanks.
so like that, whatever the value is, you can calculate. Okay, this is clear.
Yes, ma'am. Clear. And my last doubt is related to the assignment two. Assignment two. I just open the questions only. Ma'am, like in assignment class, for example, there is a one. I give one question number one in assignment two. We have all the terms like uh, 27. After the all calculation, the question is Ks value of the GD's OH3 is 2.8 into 10 ki power 23. The pH at the GDOH being to be precipitated is to be
So
using the formula GDOH power whole like downside we can write 3 and GD power 3 plus 3OH. Then after that, we can make the calculation, like make the, what do you say, make the equations. And in that equations, last what is my question is, Suppose we have the term 28 into 10 की power minus 27 divided by 27 like this. So answer is come in around always like 1.034 into 10 की power 23. So like sometimes it's difficult to me to by using that calculator. We
So
can.
yeah, I'll tell you. So uh, whenever you have, let's say, any whole number like 28 into 10 raised to the power of some big number minus 27 or like that, and divided by some other number into 10 raised to the power of minus 26. So you can just divide the whole numbers separately and calculate for the 10 powers separately. So power of 10 can be calculated. So if you have 10 raised to the power of a divided by 10 raised to the power of b, okay, do you understand this? If you have 10 raised to the power of a divided by 10 raised to the power of b, you can write it as 10 raised to the power of a minus b, okay.
Okay.
So if I have 10 raised to the power of minus 27 divided by 10 raised to the power of minus 30, okay, so that will be 10 raised to the power of minus 27 minus of minus 30, that is 20. Do you want me to write? Wait, let me try writing it. If I can. Can you open the whiteboard? Are you able to open the whiteboard? I'll just try writing it on the board.
Once again.
In the chat section, if you see messages, there will be a link. In this video call, the messages section will have the
Yeah,
link.
yeah, ma'am. Yes, yes. I'll
Yeah.
just be off.
Can you open the
Just sign
link?
it in that. When you need to access, there's a mentions access request. Request access. Can I click?
Yeah. Click.
Okay. When you have received anything, I'll put the request.
I'm checking. Okay, I can't really. Okay, I'll just share the screen.
One minute, ma'am. I'll just maybe they can
Wait, wait,
come.
wait, wait, wait. I'll just do this. Easy. You can see my screen right now, right? You can see my screen now, right?
Yeah.
Yeah. So if I have 10 raised to the power of minus 27 divided by 10 raised to the power of minus 30, this can be written as 10 raised to the power of, in the bracket you can write minus 27 minus of minus 30, okay. So now minus of minus 30 means plus 30, right? So you can write this as 10 raised to the power of minus 27 plus 30. right? Minus 27 plus 30 is
3.
okay. So this is equal to 10 raised to the power of
3.
3. So now you can solve what you were asking. What was the equation given?
28 into 10 की power 24 divided by 27.
So 10 की power 24, you can just keep it like that, and 28 divided by 27 would be, see in the calculator, 28 divided 27 is equal to 1.037. So 1.03 or 1.04 into 10 raised to the power of 24. Okay, this is clear?
Clear.
Yeah. So I'll start my class
Yes.
now. Yeah. So today is the last session of, like, in this series.
Yes.
So for the course Environmental Chemistry and Microbiology.
Ma'am, after that class, now you teach just two questions or like that.
Yeah, sure. After this whole, like, discussing all the questions of today, you can
Yeah.
ask all the questions at the end. Okay. So starting with the first question. So today we will be discussing the questions from the topics metabolic diversity, which was continued from the previous week. So we will have some other electron acceptors other than oxygen, and other than that we will study about some biogeochemical cycles. We will see what are those. Okay. So the first question is, consider consider the following with respect to carbon cycle. So carbon cycle is one of the biogeochemical cycles. Ocean, forests, soils and abandoned mines. Which of the following, which of the above mentioned, cannot act as carbon sinks? So what are carbon sinks, we will see. So among these four options, which of these options cannot act as carbon sinks? So the
Fourth.
correct, yes, fourth. So the correct answer is abundant mine. It cannot act as carbon sink. Why? Because a carbon sink can absorb more carbons from the atmosphere than it would release. So plants, oceans, soils, all of these sources can absorb carbon more than it can release. Okay. In contrast, a carbon source releases more carbon into the atmosphere than it can absorb. For example, volcanic eruption, burning fossil fuels. So if you see in all the options, ocean, forest, soils, they can absorb the carbon dioxide from absorb the carbon from the atmosphere more than it can release, and abundant mine can release more carbon than it can absorb. So abundant mine cannot act as carbon sink, it's a carbon source. Okay, I hope that's clear.
Clear.
So the next question, question number 2. What organism represents the symbiotic relationship between the roots of a plant and fungi? Okay. So option A is legume, option B is mycorrhiza, option C is blackroot, and option D is lichen. What organism represents the symbiotic relationship between the roots of a plant and fungi? Do you know the answer?
I think lynching,
No, it's pronounced as lichen. The
like
ch is pronounced as like k. Okay, so lichen, and the correct answer is mycorrhiza. So mycorrhizae, like, as plural, it is written as ZAE. If you are writing it in singular form, it will be mycorrhiza, ZA. Okay, so mycorrhizae is a symbiotic relationship between the roots of a plant and a fungus. So what do you mean by symbiotic relationship? Do you know what symbiosis is? So symbiotic relationship is basically, uh, a relationship where both the parties are benefited from each other. So if we are saying that there's a symbiotic relationship between the roots and the fungus, that means both the fungus and the roots of the plant are getting benefits from each other. So fungus is helping the helping the plants, and the plants are helping the fungus to survive. Okay, so that's a symbiotic relationship. So mycorrhiza, it's basically a fungus is associated with the root of the plant. Okay, so in that case the fungus utilizes simple carbohydrates or inorganic nutrients from soil. So if a fungus is just randomly growing in the soil, it will have to hunt for the nutrients, and it will have to find the things to eat and then break down complex substances like cellulose, leaf litter. But in this case, since it is associated with the plant roots, it can easily get these simple nutrients from the soil, and from the soil for its growth, and it does not have to break any of these complex substances. So it's easier for the fungus. So plant is in a way helping the fungus. Now how is fungus helping the plant? So what the fungus is doing? Fungus is improving the ability of the plant to absorb nutrient from its environment. So generally fungus have thin hair like or fiber like uh, tissue called mycelia. So their cells are arranged in a thread like structure called mycelia. So those fungal mycelia are associated with the root surfaces of the plant, and which is actually increasing the root surface area or the volume, and thus increasing the absorptive area of the plant root system. So basically the fungus is helping the plant to grow, plant to absorb nutrient, more and more nutrient, and the plant is helping the fungus by providing its simple substrates to grow on. So both plant and fungus are helping each other to grow. That is called a symbiotic relationship, and this particular plant and fungal relationship is called mycorrhiza. Okay, so correlation between plant diversity and abundance and diversity of the mycorrhiza. Okay, so the next question, question number 3. Oxidation of ammonia into nitrites is carried out by option A Nitrobacter, option B Enterobacter, option C Nitrosomonas, option D Rhizobium.
Ma'am, Nitrobacter.
Nitrobacter. So the correct answer is actually option C Nitrosomonas. So we will see. So Nitrobacter actually oxidizes nitrite to nitrate. In the question we have asked ammonia into nitrites. Okay, so Nitrobacter is actually doing the reverse reaction. From nitrite it is Nitrobacter is doing the nitrite oxidation. So from nitrite it is making nitrate, NO2 minus, from NO2 minus it is making NO3 minus. Okay, nitrite oxidation. The other options were Enterobacter, Nitrosomonas and Rhizobium, right? So Enterobacter, it's a coliform. It is generally present it is generally a pathogenic bacteria that causes these urinary tract infection and respiratory tract infection. So these are generally present in the GI tract also. So they cause the infections. Nitrosomonas, it can use ammonia as a source of energy, and in the presence of oxygen it utilizes CO2 as a carbon source, and so basically it can oxidize ammonia into nitrite. So the if you remember in the previous class also I have told you in the nitrogen cycle the nitrogen is fixed into ammonia. After ammonia, the ammonia gets oxidized into nitrite, that is NO2 minus, and then nitrite gets further oxidized into nitrate. So there are two oxidizing steps. So two types of oxidizing bacteria are present. One is,
Ma'am,
yes,
ma'am, can you tell once again the denitrification step of that nitrogen cycle?
yeah. So I have the slides also in the subsequent, like I have that particular slide also. But so there is difference between nitrification and denitrification. Okay, so you have nitrogen in the atmosphere. First step will be nitrogen fixation, so that nitrogen will be fixed into ammonia. Okay, now ammonia will get oxidized into nitrites, and then nitrites will get further oxidized into nitrates. This process is called nitrification. Okay,
Yes,
ammonia,
ma'am.
that is NH4, will go to NO2 minus, and then it will go to NO3 minus. So it will be nitrification. Denitrification is basically the opposite of this reaction. So from nitrates, NO3 minus, going back all the way to atmospheric nitrogen, or N2, gaseous nitrogen, that is denitrification. Okay,
Okay, ma'am.
I will show the slide. I think I have it, so I'll show it. So Nitrosomonas, Nitrosococcus and Nitrosospira, all these bacteria converts ammonia, that is NH3, into nitrite, that is NO2 minus. Okay, Nitrobacter converts NO2 minus into NO3 minus. Okay, so both are nitrification, both are involved in nitrification, but at different steps. Rhizobium, it's a genus of gram-negative soil bacteria that can fix nitrogen. So the first step of nitrogen cycle, nitrogen fixation. So it can fix the atmospheric nitrogen into ammonia, and then further nitrites and nitrates. Okay, so these kind of bacteria, they are known to form symbiotic relationships with the roots of the legumes, enabling nitrogen fixation. So you have seen in case of mycorrhiza, the fungus was associated with the roots of the plant to
Oh,
help
higher
them,
plant.
yes, to help them absorb nutrients, right? In this case, rhizobium, it's a bacteria. It is also associated with the roots of the plant, but these are special plants. These are called leguminous plants. So all these beans or pulses that you eat, those are leguminous plants. So the roots of those legumes, they have certain structures called nodules inside which these bacteria are present, and they can help fix the nitrogen from the atmosphere. Okay, so these are the process actually: nitrification, denitrification and nitrogen fixation. So nitrification, as I have told you, ammonia to nitrite and then nitrite to nitrate. There are two steps. Okay, and different organisms are doing it. Then denitrification is the reverse of this. So basically from nitrogen it is fixed into ammonia, and from ammonia it goes to nitrite and nitrate, and then the opposite, from nitrate again going back all the way to nitrogen. This is called denitrification, and you have certain examples for that. Then in case of nitrogen fixation, you have different processes: aerobic, anaerobic, symbiotic, ammonification and anammox. So you have different bacteria which are doing the same reaction, basically fixing the nitrogen into ammonia, but they are doing it via different process. Some are aerobic, they are doing it in presence of oxygen. Some are anaerobic, that means they are doing it in absence of oxygen. Symbiotic means they are associated with some other species, like for example rhizobium, it is associated with the roots of a plant species, and then they are doing this. So different process. Ammonification, again Some bacteria are just converting the nitrogen into ammonia directly. Okay, and anemox is a process where the nitrite along with this ammonia gets converted into nitrogen. So this is similar to denitrification process. Okay, like it's a part of denitrification process, and Brocadia species are generally known to do this type of reaction. Okay, is
Yes,
this
ma'am.
clear now, nitrification and denitrification?
Yes, ma'am.
So the next question, question number 4. What organism is the result of a symbiotic relationship between a fungus and an alga? So now we have seen the symbiotic relationship between the fungus and the plant, that was called mycorrhiza. So what is the symbiotic relationship between fungus and an algae called?
Lichen.
Yes, option A, lichen is the correct answer. So lichen is the symbiotic relationship between fungus and algae. Mycorrhiza I have already told you, plant roots and fungi. Rhizobium also I have told you, they are associated with the roots of the leguminous plants and they are the bacteria that are associated with the leguminous plant. So bacteria and plant leguminous roots, it is rhizobium. Fungus and plant roots, it is mycorrhiza. Fungus and algae, it is lichen. All of these are symbiotic relations. Okay. So the next question, question number 5. Which of the following is the correct reaction representing the denitrification process? So now I have explained already what is nitrification and denitrification. So you can answer.
Ma'am, it is, ma'am, the option C,
Option
nitrate to
C. Yes.
nitrogen.
So so when the word denitrification is there, that means it is, so nitrification, you remember
Reverse.
it like this. Nitrification means the forward reaction from nitrogen to ammonia, then nitrite, and then nitrate. Okay, and the reverse reaction from nitrate to nitrogen, this is denitrification. Okay.
Yes, ma'am.
Yeah. So denitrification process involves the reduction of NO3 minus, thus yielding the molecular nitrogen N2. So basically this is, the forward reaction is oxidizing reaction and the reverse reaction is the reduction reaction. So if you see the oxidation state of N here, you will see this is in positive minus 6, so minus 5, so plus 5. The oxidation state of nitrogen is plus 5 here, and the oxidation state of nitrogen here is 0. So basically
0.
this is reduction reaction. Okay, so this is the completely reduced form of nitrogen and this is the completely oxidized form of nitrogen. So when the completely reduced form of nitrogen is getting oxidized, it is called nitrification. When the when completely oxidized form is getting reduced, it is called the denitrification. Okay.
Yes, ma'am.
Question number 6. Under anoxic conditions, green and purple sulfur bacteria can utilize dash as the source of energy. Option A, nitrogen. Option B, oxygen. Option C, carbon dioxide, and option D, light. So I've told you about the phototropic and I've told you about the phototropic and the chemotrophic bacteria, right? In the last class. So do you know the difference between phototropic and chemotrophic?
Ma'am, chemotrop, it is in the presence of chemicals, and phototrop, it is the presence in the sunlight.
Yes. So if the source of energy is light, it is called phototropic. If the source of energy is chemical, it is called chemotropic. So here the green and purple sulfur bacteria, they are phototrophic bacteria. So the answer for this question is light. So they can use light to reduce carbon dioxide to carbohydrates using H2S instead of water. So that means they are phototrophic bacteria, that is they are using light as the energy source. They are reducing CO2, that means they are autotrophic. They can use carbon dioxide as the carbon source, and they are using H2S as the electron terminal electron acceptor, that means they are working in anoxic conditions without oxygen. Okay.
Yes, ma'am.
So the next question, question number 7. During anaerobic decomposition, complex polymers are broken into simple monomers in which of the following steps? Methanogenesis, acetogenesis, fermentation and hydrolysis. You have 4 options. During anaerobic decomposition, complex polymers are broken into simple monomers in which of the following steps?
Ma'am, hydrolysis.
Yes, the correct answer is hydrolysis. So you have this is the flow chart of the anaerobic decomposition. So anaerobic decomposition generally takes place in the sewage treatment plants, where the all the after the primary treatment, all the organic matter is filtered out and taken to the anaerobic sludge digester, where the anaerobic decomposition takes place. So what happens is, so the organic matter will have complex polymers of carbohydrates, proteins, fats. So these are basically biomolecules, right? So big biomacromolecules. So these biomacromolecules will be present in the organic matter. Okay. So these have
Yes,
to be
ma'am.
broken down into simple monomers. So carbohydrates will be broken down into sugars, proteins will be broken down into amino acids, and fats will be broken down into fatty acids.
Fatty acids.
Okay, so all these breaking down reactions, they take place by the process of hydrolysis. Okay, and there are certain hydrolytic bacteria that can do these kind of reactions. Once these are broken down, now they can enter the fermentative pathway. Since it is anaerobic, so only fermentation will happen. I have told you about the aerobic and the anaerobic respiration processes. So fermentation will happen. So either it can get degraded into H2 and CO2, or it can get degraded into
Propionate.
bigger C3 or C4. So CO2 means single carbon molecule, acetate means double carbon molecules, and then bigger molecules, that is
Triple.
3 carbon or 4 carbon. Butyrate is 4 carbon, propionate is 3 carbon, and so on. So if it is broken into CO2 and H2, what happens? Acetogenesis is happening, where there are bacteria called acetogens which can combine the CO2 and H2 molecules to form acetate molecules. Okay, these acetate molecules then will be converted into methane and carbon dioxide by a process called methanogenesis, and the bacteria that is involved in this process is methanogens. Similarly, these bigger molecules C3 and C4, they will be first hydrolyzed by fatty acid oxidation sorry, and they will either be converted into H2 and CO2, or they will directly be converted into acetate. If they are converted into H2 and CO2, again acetogenesis will take place like this and acetate will form. Finally, all the acetates will be converted into methane and carbon dioxide by bacteria called methanogens and by the process called methanogenesis. Okay, so there are 4 major processes that are happening: hydrolysis, then fermentation, then acetogenesis, and then methanogenesis. This is clear? All 4 processes?
Yes, ma'am.
Yeah.
Yes.
So you have to just make sure that you remember the names and what the processes are, like what is happening in each step. And if you find any example, you can remember the names of the bacteria, or at least the category of the bacteria. Like methanogenesis is carried out by methanogens, acetogenesis is carried out by acetogens, and so on. Okay.
Ma'am, in fermentation process, ma'am,
Yes.
one substrate level phosphorylation something is given. What is that?
I have told you about glycolysis, right? So in glycolysis, if you remember, from glucose it gets converted into pyruvate. So in that 10-step process, there are 2 steps in which ATP is formed. So those 2 steps are substrate level phosphorylation steps. So one is pyruvate kinase, where the phosphoenol pyruvate is converted into pyruvate, and the second is I think glyceraldehyde 3 phosphate phosphate is, I got confused in the second step, but there are 2 steps. So if you go to my previous lecture videos, or you see my slides, you will find the 10-step glycolysis process, where 2 steps are there where ATP is forming. So those 2 steps basically correspond to the substrate level phosphorylation. Okay.
Okay, ma'am.
Since one substrate is being used and the phosphorylation is taking place, so that's why it is called substrate level phosphorylation. So you have oxidative phosphorylation and substrate level phosphorylation. So in presence of oxygen, the whole electron transport chain occurs, and your ATPase works by the proton motive force, and ATP is generated. All these processes are at the end generating energy. So an energy is generated in form of ATP. So whichever step is making ATP, it is a process of generating energy, and it is a process that is important for survival for the organism. Okay.
Okay, ma'am. Archi.
Yeah, just a second. Yes, so 1,3-biphosphogylicyrate is converted into 3-phosphoglycerate. So in that step also, ATP is generated in process of glycolysis. So you have 2 steps: one where phosphoenol pyruvate is converted into pyruvate, where pyruvate kinase is working, and the second where 1,3-biphosphoglycerate is converted into 3-phosphoglycerate, where phosphoglycerate kinase is working. Okay, so these 2 kinases can generate the ATP, and that is called substrate level phosphorylation. Okay. So question number 8, methanogens belong to which of the following domains? So you answered option C, archaea. So they can produce methane during metabolism, and they belong to the archaea domain. What is the recommended period of time in terms of days for a BOD
5
test?
days.
Days.
Yes. So the recommended period of time for the standard BOD test is 5 days according to the APHA, AWWA and WEF. In India, the BOD discharge standard is based on 27 degrees Celsius for 3 days. Okay. So the correct answer is option B, 5 days. Question number 10. Dash is the largest source of SO2 in the atmosphere. Option A, burning of fossil fuels. Option B, locomotives, ships and vehicles. Option C, volcanoes. And option D, extraction of metals from ores.
Ma'am, A.
Ma'am, option D.
So the correct answer is option A, burning of fossil fuels. So all the other three option in locomotives and ships and vehicles, you know what is being emitted? Like its main source is carbon monoxide. Okay. So SO2 source of SO2 is burning of fossil fuels. So the next
Ma'am, in
question.
previous question, in extraction of the that metal from ores, in that
Yes.
step also the SO2 is coming now.
Yes, but it's not the largest source. In the question you have been
Okay.
asked, the largest source. So you have to see that. Okay.
Okay.
Because that's not the most common process, extraction of metals from ore, but burning of fossil fuels everyone is doing. Metal extraction from ores is only happening at the industrial levels, but fossil fuel burning is happening in almost every household, right? So that is the
Yes, ma'am.
largest source of SO2.
Okay.
Question number 11. SO2 released into the atmosphere is converted to dash and dash. So SO2 is finally converted into two forms. Option A, sulfur and oxygen. Option B, sulfuric acid and sulfates. Option C, hydrogen sulfide and oxygen. And option D, sulfonic acid and oxygen.
Ma'am, option A.
Ma'am, option
Option
B.
Option B is the correct answer. Option A is sulfur and oxygen. So sulfur dioxide, when it is released in the atmosphere, it will be converted into sulfuric acid and sulfates. Basically, it is getting oxidized. Okay. Question 12. Biogas generated at the end of the anaerobic digestion is mainly composed of dash and dash: ammonia and methane, H2S and CO2, CH4 and CO2, CH4 and CO, that is carbon monoxide.
Option A.
Ammonia and methane. Biogas generated at the end of anaerobic digestion. So I have already told you, right? Anaerobic digestion, I have explained the whole process. So what were the two products that were getting released? Methane and CO2. So I had shown you this pathway, right? We have discussed hydrolysis, fermentation, acidogenesis and methanogenesis. So this is the basically anaerobic decomposition or anaerobic digestion. So where the final step, methanogenesis, is happening, and methane and carbon dioxide are the main gases that are getting released. Okay, here. So from complex polymers all the way to methane and carbon dioxide, these are called the
Okay,
biogas.
ma'am.
Okay.
Yes, ma'am.
So question number 13. Which enzyme can catabolize starch? Option.
Amylase.
Yes.
Amylase.
Amylase is the correct answer. Cellulase, dextranase, chitinase and amylase. So cellulase breaks down cellulose, dextranase breaks down the polymer of dextran, chitinase breaks down a substance called chitin. Chitin is generally present in the fungal cell wall, and amylase breaks down starch. So amylase it catalyzes the hydrolysis of starch into simple sugars. Starch is a straight chain polymer of glucose with alpha 1,4 glycosidic bonds between individual D-glucose molecules. So if you see, between the glucose molecules there is alpha 1,4 glycosidic linkages. This CH, this kind of linkages are called glycosidic linkages, and these are broken down by the enzyme called amylase, and this breakdown is the process of breakdown is called hydrolysis, and single molecules of glucose are released after the hydrolysis. So from the straight chain polymer of glucose with alpha 1 4 glycosidic bond, it is hydrolyzed into simple D-glucose molecules. Okay, like this. So you have the starch molecule and you have the single glucose molecule after hydrolysis. So question number 14, fatty acids are metabolized through which of the following process? Alpha oxidation pathway, beta oxidation pathway, hydrolysis reaction and oxidative phosphorylation.
D.
D. Oxidative phosphorylation. I have just told you what oxidative phosphorylation is. I have discussed it in the previous classes also. And I have discussed the fatty acid metabolism also in the previous class. So the answer for this question is option B, beta oxidation. So oxidative phosphorylation, what is oxidative phosphorylation? I have just told you. Tell me.
Ma'am, oxidation is in the presence of phosphor.
Oxidation in presence of phosphor. What is phosphor?
And PO4. Phosphate, ma'am.
That is oxidative phosphorylation. I just told you about electron transport chain. You asked me about substrate level phosphorylation. I have discussed about electron transport chain in very details uh, in I think 9th problem solving session. So in oxidative phosphorylation, what is happening? It's basically the complete oxidation of the glucose into CO2 and H2O. Okay, and in that process ATP is generated. So the term phosphorylation is used because the phosphate groups are added to the ATP. So ADP plus phosphate becomes ATP. So that is the phosphorylation process, and the term oxidative is there because it is happening in presence of oxygen. Okay, it's not oxidation of phosphates. Okay, it's oxidation of ADP into ATP, you can say, or it's phosphorylation of ADP into ATP. It's oxidation of glucose into CO2. Beta oxidation pathway is the pathway that by which fatty acids are metabolized. So basically fatty acids are long chain carboxylic acids. So those carboxylic acids at the end, they will have the COOH group. So one coenzyme A replaces the OH or the H2O part of it. So it replaces the OH minus and coenzyme A is added, and this coenzyme A, once the coenzyme A is added, it is very easy for the enzymes to work on this molecule, and it can break it into acetyl coenzyme A. And this acetyl coenzyme A, you can know, it can enter which pathway? Acetyl coenzyme A can enter which pathway?
Glycolysis.
From where it will enter the glycolysis? What is glycolysis? What is glycolysis?
Ma'am, acetyl CoA was the first in the first process
Of glycolysis.
of that cycle. Ma'am, glucose is converted into acetyl CoA.
Glucose is converted into
Ma'am,
acetyl CoA
acetyl CoA.
by glycolysis.
Yes,
And
ma'am.
okay, and acetyl CoA is enters which pathway?
Ma'am, I forgot that.
So we have discussed, yes. Which pathway does acetyl coenzyme enters? So we have
Ma'am,
discussed,
uh, ma'am, you tell something like that. Ma'am, monooxygenase N is converted to all in the presence of monooxygenase, and
that
all
is
is
the
converted to
part of,
NAD plus
yes, that is the
and
part of beta
N.
oxidation. That is the part of beta oxidation. We are talking about acetyl coenzyme A right now. So you have glycolysis where glucose is converted into pyruvate. I've just told you. You asked me about the substrate level phosphorylation, and I've just told you. Please pay attention to the statements that have been said in the class. So glucose is converted into pyruvate. It's a 10-step process. Okay, that pathway is called glycolysis. After pyruvate, the pyruvate gets converted into acetyl coenzyme A, and so that is the pathway for carbohydrates. Similarly, you are when you are eating apart from the carbohydrates, you are eating fats and you are eating proteins, right? So now proteins will be broken down into amino acids, and fats will be broken down into acetyl coenzyme A again, and those amino acids will also be broken down into acetyl coenzyme A. All these acetyl coenzyme A, they will enter the tricarboxylic acid cycle, TCA cycle, or the Krebs cycle, or the citric acid cycle. There are many names for that cycle, so you should remember that. So first pathway that is there in the whole aerobic respiration system is glycolysis. The second pathway that is there is the citric acid cycle, and then after citric acid cycle, all the NADH that are generated, they are
ATP
cycled to,
formation by
yes,
oxidation phosphorylation.
yes, so they are cycled to the electron transport chain, where the electrons are transported and the proton motive force is generated, and because of this proton motive force, the ATPase can now generate ATP by adding a phosphate molecule to
Form ADP.
ADP. It can add phosphate to ADP to form ATPs. Okay, so that is the whole complete oxidation of glucose into CO2 and H2O. So these fatty acids, they are broken down into acetyl coenzyme. So now one, so let's say it's a 10 carbon chain. So from that 10 carbon, 2 carbon will be released and remaining 8 carbon will be there. And then let's see how many carbons are this. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14. So it's a 14 carbon chain. So from 14, 2 will be released, acetyl
12.
CoA. So that then 12 will be remaining. Then from this 12, again same reaction will occur, and then again one acetyl coenzyme will get released and 10 carbon chain will remain, and then from that 10 carbon chain 8 will be remaining, then 6, like that. By repetitive cycles of this process, many acetyl coenzyme A will be released, and all these acetyl coenzyme A will keep entering the citric acid cycle, and during this process 5 ATP molecules are generated. Okay, so this is the beta oxidation of fatty acids. Okay,
Okay, ma'am.
so question number 15, is the following sentence true or false? Fatty acid metabolism is an ATP utilizing process. I have just told you. Now tell me the answer. I have shown you the pathway. Was it utilizing ATP or was it making ATP?
Ma'am, it was making ATP.
Yes, it was making ATP. So the statement is false. So if you see in this pathway, it is generating 5 molecules of ATP for doing this whole process, for doing this whole cutting process. So 5 ATPs are generated. So it is not a ATP utilizing process, it's a ATP generating process. Okay, so the statement is for fatty acid metabolism does not utilize ATP, instead it generates ATP. Question number 16. The range of total Gildan nitrogen in sewage is normally around option A 10 to 15 mg per liter, option B 20 to 80 mg per liter, option C 30 to 50 mg per liter, and option D 5 to 20 mg per liter. The range of total Gildan nitrogen in sewage is normally around. The answer is
Ma'am, 20 to 80.
yes, 20 to
B
80.
option.
Yes, 20 to 80 mg per liter. That is the correct answer. So, what is this total Gildan nitrogen? It is basically, so Gildan process, it has been named after the scientist. So, it's a process of determining the amount of nitrogen. So, amount of nitrogen, it is determined from, so in the sewage the amount of nitrogen is determined from the ammonia and the
organic matter which is
yes.
present in that sewage plant.
So most of the organic matters, they are composed of carbon, hydrogen, oxygen and nitrogen, right? So any organic matter having nitrogen, that will be estimated, and other than that whatever ammonia content is there, so after nitrogen fixation ammonia is present, right? So whatever ammonia content is there, so from that nitrogen will be estimated. So so the total Gildan nitrogen comprises of the nitrogen coming from the organic matters plus the nitrogen coming from the ammonia, and it is determined by the Gildan process. That's why it is called total Gildan nitrogen, TKN. Okay, and it is normally around 20 to 80 mg per liter. Question number 17. According to the new National Green Tribunal guideline, what is the maximum BOD value at 27 degrees Celsius for treated wastewater being discharged into an inland surface water body? So, what is the maximum BOD3 value at 27 degrees Celsius for treated wastewater being discharged into the inland surface water body?
30.
The correct answer is 10 mg per liter. So 10 mg per liter is actually the permitted amount. So 10 mg per liter is the revised sewage discharge standard for wastewater being discharged in mega and metropolitan cities. In India, mega cities are defined as the cities with population greater than 10 million, and metropolitan cities are defined as population greater than 1 million. So you can see in this chart also the BOD value is given. So for mega and metropolitan city, 10 is the value, 10 mg per liter. For class 1 cities that have a lesser population than these two, like lesser population than 1 million, class 1 cities, they have the applicable limits to 20 mg per liter. Other cities have 30 mg per liter, and deep marine outfalls, they also have 30 mg per liter of BOD value. Okay. So question number 18. The ruminant digestive system is analogous to option A aerobic digestion tank, option B anaerobic digestion tank, option C trickling bed reactor, and option D continuous stirred tank reactor. So what do you mean by ruminant digestive system?
Ma'am, digestive systems of like cows, buffaloes.
Yes. So they are analogous to.
Ma'am, actually, I am not able to understand this trickling bed reactor.
Uh, so it's a bioreactor actually. So it's a bioreactor where you have like there are different types of bioreactor. We'll have to go in deep to understand that. I don't think that's the scope of this session. That's in the scope of this session. So here the answer is between anaerobic and aerobic. Actually, the answer is
Ma'am, anaerobic.
anaerobic digestion tank. So in the rumen, so rumen means the gut of the stomach of the.
Cows, buffaloes.
Yes. So they have actually 4 compartments, and in that, uh, in the rumen of those animals, there are certain anaerobic bacteria that are present that can actually digest and form. So you know, cows and buffaloes and all these ruminating animals, they eat uh, dried grass and stuff like that, right? So those are
Yes.
mostly plant material. So those plant material are rich in cellulose. So now those complex plant material, they have to be digested. So how they are digested? They are digested by the anaerobic bacteria. So I've told you about the anaerobic.
And they have the particular enzyme to digest that cellulose material
uh,
present.
Not just the enzymes. So exactly, that's what I'm saying. The animals don't have the enzymes. The animals have these anaerobic microbes inside their gut that can actually metabolize these complex cellulosic materials. So you have seen the anaerobic decomposition, right, where the carbohydrates, proteins and fatty acids were hydrolyzed, and then
Yes,
fermentation
ma'am.
was
Yes, ma'am.
happening, and then acetogenesis, and then finally methanogenesis was happening, right? So
Yes, ma'am. Yes,
similarly,
ma'am.
similar process happens in the gut uh, of these animals. uh, in the gut of these animals, anaerobic microbes are present, and they can break down these plant materials and by hydrolysis and other steps, and then finally methanogenesis happens, where carbon dioxide and methane are released. So if you have seen the dung of these animals, cow dung and buffalo dung, those are actually used as fuels in rural parts of India. If you see the gobar that is produced by these animals, it is used as fuels. They dry the cakes of these uh, dung,
Yes,
and
ma'am.
they use it as fuel. So it's a great fuel source because it is rich in carbon dioxide and methane. Okay, and that cow dung is actually used for biogas production also. So carbon dioxide and methane, I have told you, these are mainly the biogas. So this the digestive system of these animals, it is very similar to the anaerobic digestion tank that is present in the sewage treatment plant, where anaerobic decomposition happens. Okay, this is clear.
Yes, ma'am.
Okay, so next question: which of the following sources is responsible for the most amount of methane emitted globally? Which of the following sources is responsible for the most amount of methane emitted globally? Coal mining, rice cultivation, wastewater, and enteric fermentation.
Ma'am, what is enteric fermentation?
Yeah, so the answer for this question is enteric fermentation. I'll tell you what is enteric fermentation. So enteric fermentation is actually what is happening in the gut of these ruminant bacteria. So ruminant animals. I'm sorry. So enteric fermentation is the part of the digestive process in the ruminant animals where the microbes in the digestive tract decompose and ferment the food, producing methane and methane as a byproduct. So the question was, which of the following sources is responsible for the methane emission, right? So by coal mining, rice cultivation and wastewater, methane is not emitted. Enteric fermentation, it's basically the process of this ruminant digestion, and that can generate methane and carbon dioxide as byproduct. Okay.
Okay, ma'am.
So question number 20. Which pair of enzymes catalyze oxidation of hydrocarbons under aerobic conditions? Lipase and amylase, cellulase and hydrogenase, monooxygenase and dioxygenase, and ligase and lipase. Which pair of enzyme catalyze oxidation of hydrocarbons under aerobic conditions?
Ma'am, monooxygenase and dioxygenase.
Yes, the correct answer is monooxygenase and dioxygenase. So you were right now mentioning that I have told you about the oxidation using these monooxygenase and dioxygenase, where the alcohol and then the aldehyde was forming from the carboxylic acid, right? So that is the
Yes, ma'am.
process. So from carboxylic acids and all these so hydrocarbons are basically the organic carbon chains, right? So it can be either aliphatic or aromatic. Okay, so like this is an aliphatic molecule, so N-octane. So it's a 8 carbon molecule without any oxygen or anything. So if a bacteria has to hydrolyze this or metabolize this, so now they have to convert it into something which can be used up easily. So N-octane will be converted into octanol by monooxygenases. So monooxygenase, by the name you can see mono and di, two names are there, right? Monooxygenase and dioxygenase. So monooxygenase adds one oxygen and dioxygenases can add two oxygens at the same time. So if you see here, by monooxygenase one oxygen is added, so it was CH3, now one oxygen is added, so it is CH2OH and it is becoming octanol, an alcohol. Okay, then after this alcohol is converted into aldehyde, and then the aldehyde is converted into the carboxylic acid, and this carboxylic acid is nothing but the fatty acid, and you have seen how fatty acids are metabolized by beta oxidation, and you had seen in the previous slide only. So fatty acid will be broken down into acetyl coenzyme by 2 carbons. Okay, so
Ma'am, what is AMP?
adenosine monophosphate. So ATP is adenosine triphosphate, then ADP is adenosine diphosphate, this is monophosphate. So 2 phosphate molecules, so this is PPI means pyrophosphate. So 2 phosphate together are getting released and AMP is forming.
Okay, ma'am.
Okay. So here also if you see, this is an aromatic hydrocarbon. So it's a benzene ring, right? So benzene.
Ma'am, please repeat the last one once, ma'am. The last. That cycle, ma'am. Yes, ma'am. Can you please repeat once that cycle?
Yeah. So so hydrocarbons means a chain of carbon and hydrogen, right? So it can either be a straight chain or it can be a aromatic chain with double and triple bond. Okay. So if it's a straight chain with single bonds, it can be an octane or higher, like it's a 8 carbon molecule, it can be a 9 carbon molecule, 10 carbon molecule, or any n number of carbon molecules, right? So it can be an alkane. That alkane will be converted into alcohol by monooxygenases. So monooxygenases are adding one oxygen at a time. So one oxygen is added and the alkane becomes alcohol. Then that alcohol is converted into aldehyde and then finally to carboxylic acid. This carboxylic acid is nothing but fatty acid. So I have shown you in the fatty acid hydrolysis. So this was basically the carboxylic acid. In place of OH, the coenzyme A will get attached and then it can get cleaved by the beta oxidation pathway into acetyl coenzyme A. So if it's a 8 carbon molecule, 6 carbon molecule will be remaining, 6 carbon carboxylic acid will be remaining, and 2 carbon will be released in form of acetyl coenzyme A.
Ma'am, I am having one doubt. Why it
Yes.
is cutting only on the two chain, like two
The
bond is
process is
cutting?
like that only, like the reaction goes in that way only. So acetyl coenzyme A is the byproduct. So if it cuts here, at the end it has to make acetyl coenzyme A, then only it can enter the pathway, right? Enter the subsequent pathway. So the nature has made the system like that. Even if it is cutting here, let's say it will cut here, at the end all of these carbons will be chopped off in form of 2 carbon molecule, that is acetyl coenzyme A. So acetyl coenzyme A is actually 2 carbon molecules. Okay.
Okay, ma'am.
Okay. So from the octanoic acid or whatever carboxylic acid is there, it will get converted into acetyl coenzyme A, and acetyl coenzyme A can then enter the pathways. So this is oxidation of aliphatic hydrocarbons. If you have aromatic hydrocarbon, so aromatic hydrocarbon means it has double bonds. So you have a benzene ring, let's say. So benzene ring will get oxidized by monooxygenase. So one oxygen will be added and it will form a epoxide bond. So benzene epoxide. This benzene epoxide will then get converted into benzene diol, like an alcohol, and then that alcohol will get converted into a catechol, that is 2
OH.
OH on the benzene ring at the ortho position. So this catechol, now catechol will then get converted into catechol, so basically it's cis muconate by catechol dioxygenase. So here the purpose is basically to break down this aromatic compound. So this is a aromatic hydrocarbon. So we have to, like, whatever bacteria is growing on this benzene, using this benzene as a carbon source, it will have to break the carbon so that it can utilize. So for first they have to break the ring. So after catechol, now the ring has to be broken. So for that, a dioxygenase molecule adds 2 oxygen. The dioxygenase molecule adds 2 oxygen. The dioxygenase molecule, it will add 2 oxygen in the catechol, and then the rearrangement will happen and the compound will, this catechol will be broken into cis muconate. Okay. Similarly, for the toluene molecule, a dioxygenase will add 2 oxygens to make it methyl catechol. So it is a catechol molecule with a methyl group on it. Okay.
Yes, ma'am.
And then from methyl catechol, it will again get oxidized by a methyl catechol dioxygenase into this compound. Okay, so that's how monooxygenases and dioxygenases catalyzes the oxidation of the hydrocarbons, both aliphatic and aromatic hydrocarbons. Okay. Okay. So, question number 21. During the lightning of high temperature, during lightning, the high temperatures and pressures created in the air converted nitrogen into dash. Option A, chloroform. Option B, ammonia. Option C, oxides of nitrogen. And option D, oxygen. So, nitrogen is converted into what? By lightning, nitrogen is converted into
Ammonia.
ammonia.
uh, so nitrogen is actually finally converted
Oxides
into
of
oxides
nitrogen.
of nitrogen, right? So in
Yes, ma'am.
this nitrogen cycle, so I have told you atmospheric nitrogen, it is fixed first into ammonia. So this ammonia
Ma'am, that will be
fixation,
nitrite and nitrate, na oxides of nitrogen.
yes, oxides of nitrogen means nitrite and nitrates. So basically what happens, atmospheric nitrogen, it gets fixed into two paths, like it gets fixed by two processes. One is by bacteria. So I've told you about rhizobium. It was present in the roots of the leguminous plants. So it's a leguminous plant and the roots have the nitrogen fixing bacteria. So they can fix the atmospheric nitrogen, and they can, all these atmospheric nitrogen can be converted into ammonia or ammonium ions. This process is called ammonification. Along with that, lightning, since lightning has high temperatures and pressures, so like during lightning, high temperatures and pressures are created in the air. it it can convert nitrogen into oxides of nitrogen and dissolves in the water and comes down along with rain. So when lightning is happening during rainfall, the high temperature and pressure that is created in the air, they actually convert the nitrogen in the air into oxides of nitrogen, and that those oxides of nitrogen, basically nitrites and nitrates, they can get dissolved in the rainwater and can fall on the soil. So now the soils have nitrites and nitrate that came along with the rainfall and lightning, and along with that I have told you about nitrification, that was conversion of ammonia into nitrites and nitrates. So by those bacteria, so there were nitrification,
Also,
ammonia oxidizing bacteria and nitrite oxidizing bacteria. So ammonia oxidizing bacteria was nitrosomonas and nitrite oxidizing bacteria was nitrobacter.
nitrobacter.
Okay, so all those, finally all these atmospheric nitrogen, they will get converted into nitrates. That's the final oxidized product. After nitrates, now it has to go back to the atmospheric nitrogen. So denitrifying bacteria will play the role in that. So they will convert the, so basically they will reduce the nitrates into atmospheric nitrogen. Apart from that, some assimilation steps takes place where it gets assimilated in the plants, and then other bacteria can use these
Okay,
as
ma'am.
the nitrogen source. Okay,
Okay, ma'am. Yes, ma'am.
so this is the complete nitrogen cycle. Okay, so here
Yes,
you
ma'am.
can I think now the process of nitrification and denitrification is clear. Ammonification is basically nitrogen fixed into in form of ammonia. This
Ammonia.
process is basically carried out by bacteria. Okay, and other than that, lightning can convert nitrogen into nitrites and
Oxides of
nitrates.
nitrogen.
Yes, nitrites and nitrates that falls on the soil along with the rainwater, and in the soil now all the nitrites and all the ammonia are finally converted into nitrates, and this nitrate goes back to atmospheric nitrogen by denitrification process. Okay.
Yes, ma'am.
So question number 22. Which of the following is a sedimentary type of biogeochemical cycle? Phosphorus and carbon monoxide, oxygen and nitrogen, phosphate and nitrogen, and phosphate and sulfur. Which of the following is a sedimentary type of biogeochemical cycle? Any idea? So there are two types of biogeochemical cycle. One is gaseous and one is sedimentary. Now can you tell which one is gaseous and which one is sedimentary? Sedimentary means it is present on the soil surface or the earth surface. So which cycle is happening in on the sediments and which cycle is happening on in form of gases?
Ma'am, phosphorus and carbon monoxide happens.
Carbon monoxide is a gas or a
Ma'am, gas.
so how it is a sedimentary type? Okay, carbon monoxide is a gas, so it is not a sedimentary type. Oxygen, nitrogen.
Ma'am, phosphate and sulfur is a sediment type of
Yes,
bio.
phosphate and sulfur is the sediment. So you have to think, like you see all the options. Now you know oxygen and nitrogen and carbon monoxide. All these three are gases. So remaining option is phosphate and sulfur. So these are the sedimentary type of biogeochemical cycle. Okay.
Yes, ma'am.
Question number 23. Aerobic oxidation of H2S is carried out by which of the following bacteria? Purple sulfur bacteria, green sulfur bacteria, thiobacillus and Beggiatoa, or D-sulfovibrio and D-sulfobacter? Aerobic oxidation, you have to pay attention. Aerobic oxidation is carried out.
Purple sulfur, purple and green.
Purple and green sulfur bacteria, I've we have discussed about this. It was happening aerobically or
Aerobic, yes,
anaerobically?
ma'am. Ma'am, aerobic.
It was happening aerobically. It was happening anaerobically. So purple and green sulfur bacteria can actually use the
Ma'am,
H2S.
sorry, ma'am. Option C, Theobacteria.
Yes, thiobacillus and Beggiatoa. Okay, that is the correct answer. So purple and green sulfur bacteria, they can actually use the H2S as the electron acceptor, terminal electron acceptor. So I'll show you the pathway. So this is the sulfur cycle. So if you see, you have sulfates, you have H2S, you have elemental sulfur, and then sulfur dioxide. Sulfur dioxide is released from certain sources. Burning of fossil fuels, we have discussed that it's the major source of SO2 emission. Then SO2 is converted into H2SO3, sulfuric acids and sulfates. That also we have discussed. SO2 is converted into sulfuric acid and sulfates. Now that sulfate, it will get assimilated in certain proteins. So you know amino acids like cysteine and methionine have SH groups. So some sulfate will be assimilated. Some sulfates will be reduced to H2S by sulfate reduction process that is carried out by D-sulfovibrio and D-sulfobacter. So these are the bacteria that are reducing sulfates into H2S. So if you remember, in the Vinogradsky column that I have discussed in the last class, if you see in the previous class slides, you will find these D-sulfovibrio and D-sulfobacter, they were present at the upper part of the column. sorry, at the lower part of the column, at the bottom of the column, and they were converting these sulfates into H2S, and this H2S was getting, since it's a gas, it was getting released at the upper part of the column, right?
Yes, ma'am.
So that Vinogradsky column is actually a representation of the sulfur cycle in a laboratory setup. Okay, so sulfates are reduced into H2S by desulfovibrio and desulfobacter. Then sulfates are assimilated into proteins, I have already mentioned. Then this H2S, now this H2S can get converted into elemental sulfur by two processes, aerobic and anaerobic. So that also we have discussed in the Vinogradsky column. So aerobic process, that was happening at the top of the column, it was carried out by bacteria like Thiobacillus and Beggiatoa. So H2S is converted into elemental sulfur. Elemental sulfur again can get oxidized into sulfates by certain microorganisms, and the H2S was getting oxidized into elemental sulfur by anaerobic process, which is present in the middle part of the Vinogradsky column. If you remember, the middle part of the Vinogradsky column had the purple and the green sulfur bacteria that were doing the oxidation of H2S. Basically, they were using H2S as the terminal electron acceptor and converted into elemental sulfur. This elemental sulfur anaerobically is also oxidized, reduced into H2S. So here only oxidation is taking place. H2S is getting oxidized into elemental sulfur. Here in anaerobic process, both oxidation and reduction is taking place. So as you can see, this is sulfate reduction. Sulfate reduction was happening by desulfovibrio and desulfobacter, whereas sulfur reduction, sulfur is reduced into H2S, this is happening by desulfuromonas. Okay, so H2S is oxidized into elemental sulfur anaerobically by purple and green sulfur bacteria, and the elemental sulfur is further oxidized into sulfates by these bacteria. But there are bacteria that can reduce elemental sulfur into H2S under anaerobic condition. Under aerobic condition, this process is not possible, but under anaerobic condition, desulfuromonas can do this reaction as well. Okay, so this is the whole sulfur cycle. This is how the sulfur is getting recycled in the atmosphere. Okay,
Yes.
so this was the last question of this session. You all are free to ask any questions now. If you have any question from the previous lectures also, you can ask, since this is the last session of this course. So you can ask me any doubt you have so that I can clarify. Do you have any question?
Ma'am, in week first acid basis salt, I am getting doubts in getting the pH questions of that.
Okay, can you tell me any particular question so that I can help you, or any particular part where you are getting the doubt? Hello.
Ma'am, I have a doubt of question like assignment to the question number 15.
Assignment 2. Okay. Ria, you were asking a question from assignment 1.
Yes, ma'am. Yes, ma'am. I'm saying that.
Okay. By the time I'll clarify Remika's doubt. Uh, so Remika, you are asking question number, which question number?
Ma'am, 15. If the solubility product KSP of the solid bio bromides is a 5 into 10 ki power 12 13 at 25 degrees Celsius. Calculate the approximately solubility of mole weight AgBr is 18.7.77. Ma'am, is there after the solving that questions I'm not getting answer actually, that's why. So,
What is the problem? You can.
yes, ma'am. The problem is that, ma'am, like there is a mentions the value of s square is given 5 into 10 ki power 12. Then after that, when we go for like half of something, like there is a mentions 5 into 10 ki power minus 13. Then we divided by half by 2, 1 by 2, sorry. And then, ma'am, how we get the answer? There is a mention 7.07 and 10 power minus 7 m. That I'm not understand how the answer will become.
Uh, just a second. Can you? I can't find the question actually which you are asking. Solubility product is given, right? Hello. She left the chat. Ria. Hello, Remeka.
Yes, ma'am.
Yeah. Yeah. So can you tell me the? Can you tell me the doubt again? I did not understand.
Ma'am, if you want, then I can show the questions. Actually, the assignment too. If you want, can I open?
Yeah. You can. I think
Just
you can share your slide. You can share your screen. I'll stop presenting. You can share your screen if you want.
yes, ma'am. Just a minute. I'm just opening the questions. Yes, ma'am. I'll just share the screen. Ma'am, have you seen? Have you able to see, ma'am?
Yes, yes.
Ma'am, the question 15, like I have a doubt, like how they get the answer of 7.07 minus 7, 10 to power minus 7 m molarity, like that I'm not understand. After calculating 5 into 10, 10 to the power 13, like that how we get it? They can do the root of 5 into 10 power
Wait,
13.
wait, wait. Uh, you are asking for question number 15, right?
Yes, ma'am.
So in question number 15, s square is 5 into 10 raised to the power of minus 13.
Yes, ma'am.
So you have to take the under root of 5 into 10 raised to the power minus 13. So basically, root of 5 is, square root of 5 is,
I have to do, ma'am, just a minute.
you can calculate in the calculator, right? So this is the calculator you have. Uh, if you see the scientific calculator, yes.
Like, ma'am, if we do the 5
So
into
write 5. So in this calculator, if you maximize, you will get the square root or x raised to the power of 1 by 2. You have to do 5 raised to the power of 1 by 2. So just, you have the square root option, right? So see, under root of x is there beside the left bracket.
this
From,
one, ma'am.
just delete everything.
Okay.
just delete everything, write 5,
This one.
just write 5, yes, this one. So just write 5 and then click on that. Delete everything. Ramika,
Then
just listen to me first. Yeah, just write first write 5, first write 5, yes, and then click on that, then click on that, yes. So this is the value of root 5. This is how you use a calculator on screen. Okay, so this is the value of root 5. So 2.23 is the value of root 5. Okay, so 2.23. Now you have to take the square root of 10 raised to the power of minus 13. So you can what you can do is you can divide it into two parts. So you can write it as 10 raised to the power of minus 1 into 10 raised to the power of minus 12, right?
okay, like we multiply it first, then after that.
So 10 raised to the power of minus 13 can be written as 10 raised to the power of minus 1 into 10 raised to the power of minus 12, right?
Yes, ma'am. But the problem is that if we put the minus value, like what happened, that is gone.
You
So,
can't write everything on the calculator. Just listen to me first. What I am saying is minus 13 can be written as minus 12 minus 1,
hmm,
right?
yes.
So if you write 10 raised to the power of minus 13 as 10 raised to the power of minus 1 into 10 raised to the power of minus 12, okay, that can be written as that.
Okay, like first, ma'am, we do minus 10 raised to
Don't
power.
write it on calculator, first write it on your notebook. Just write 10 raised to the power of minus 13 is equal to 10 raised to the power of minus 1 multiplied by 10 raised to the power of minus 12, and tell me if you understand that.
Yeah. Yes, ma'am, I understand. If we calculate, this is equal to minus 13, 10 to power
Yes,
minus 13.
yes, that you understand.
Hmm.
Now if you take this, if you have to calculate the square root of 10 raised to the power of x, okay, that will be basically equal to 10 raised to the power of x by 2. Okay, so if I have 10, let's say I have the value as 10 raised to the power of 4, and I have to take the square root of 10 raised to the power of 4. So it will be equal to 10 raised to the power of 4 divided by 2. So 4 divided by 2 is 2. So it will be 10 raised to the power of 2. Do you understand this?
Yes, ma'am.
So now tell me what will be the square root of 10 raised to the power of minus 12.
We'll do by this, ma'am, calculator, no?
No, you tell me. I told you how to calculate the square root without using the calculator for powers of 10. If 10 raised to the power of 4, if the square root of 10 raised to the power of 4 is equal to 10 raised to the power of 4 divided by 2, that is 10 raised to the power of 2.
Okay, fine. Then
Now
12
tell me,
divided by, ma'am, 2 then.
yes,
Then
so 10 raised to the power of minus 12,
it's equal to minus 6.
yes, so square root of 10 raised to the power of minus 12 will be 10 raised to the power of minus 6,
Correct.
correct?
Yes, ma'am. And that's why it's called minus 7.
Yes, and then 10 raised to the power of minus 1,
It's same as like we 1 by 2, ma'am. If we divide it by like
yes,
the minus when we either we take same as it is or either we divide by 2.
yes, it's the same thing. If you want to do this on calculator, I'll tell you how to do it on calculator as well. So just delete everything from your calculator first. Okay,
Yeah.
write 10, click on that x to the power y, click on the left bracket, Now write minus 13. Okay. And now click on the square root. Okay. So you can see it's showing it in the decimal format, but if you have to calculate in terms of, so you can count the number of, I think you have
I got
done
it. Yeah,
something
I got
wrong,
it.
but then
Yeah, I got it.
huh. So this is
Ma'am,
how you,
you can say that the 5 root, but this answer is wrong. Like 7.07 is not be there now. Like that is correct. That the Z 7.07.
uh, s square is given as 5 into 10 raised to the power of minus 13. So the square root of 5 this is equal to, yes, I think it is wrong.
Okay, fine, ma'am. Ma'am, the 16 questions here have the same doubt.
Yeah. What is the doubt?
Ma'am, after that, the like 1.4 into 10 to power 16 27. Though we have to be
So you first divide 1.4 divided by 27.
uh, okay. And then we go for, ma'am, roots, like we do for the roots, like if we go divide by, okay, I'll do it
So
once
you
more.
have to take, either you take 2 times roots, or you can do, uh, you don't have the option here.
Okay, then we do
You
root.
have the option in the scientific calculator provided by NPTEL. So you can directly write 4 there.
Yes, ma'am, I have the scientific calculator that
Yes.
also.
So if you see the uh, button next to the tan inverse,
That one, like first we do that,
first you
first we
one,
divide it,
yes, from 1.4 divided by 27 equals,
and then we do the root
okay,
of that.
don't take the square root, you have to take 1 by 4, right?
Yeah, then 1 by 4, where
So
is it?
so I'm telling you, one,
Oh,
first divide it 1.4 divided by 27.
yes, ma'am.
Then click on this button at the, in the last row, the fourth button beside tan inverse.
This one,
I can't see your arrow.
that
Beside,
one,
below that, below that, yes, yes, click on that, okay, and write 4.
4.
Just type 4. Okay, that's not what, wait, I'll see. Yeah, click on equal to, yeah, you
Yes,
got
ma'am.
the answer as 0.477, right? So what's the answer in PDF?
Ma'am, answer is 4.69.
So it's fine, na, 4.7.
Yeah. No, no,
Approximately
answer
only.
is not this one. 5.2, sorry. 5.2 in 10 to power 6.
Okay, 5.2. Go to your calculator again. Yeah, so you are getting 0.477. Now you have into 10 raised to the power of minus 16 to the power of 1 by 4, right?
Yes, ma'am.
Go to the question again. 10 raised to the power of minus 16 to the power of 1 by 4. So minus 16 divided by 4 will be minus 4. Hello.
Yes, ma'am, I'm listening.
Yeah, so minus uh, so it will be approximately equal to 5. I think because you are taking the complete decimal places, that's why you are getting this answer.
Okay.
Just a second, let
Ma'am,
me
in up
check.
that is 1.4 into 10 to the power minus 4 upon 27. How
Minus
will you?
minus 16 upon 27.
No, ma'am, when we'll solve that 10 minus 16, you have tell na that upon 2 we'll do.
So first you will have to you can't solve separately, right? You have to solve everything together. So you have 1.4 into 10 raised to the power of minus 16 divided by 27, right? Inside the bracket,
Yes,
right?
ma'am.
So first we are solving 1.4 divided 27. So you are getting the answer as 0.05. Okay? Now just write 0.05 and take the fourth root of it.
I'll do it again, ma'am, once again. Like,
Ma'am, actually, I'm not getting how to do in that calculator. I'm getting so much difficulty to that.
Uh, Remika, if you stop sharing, I'll show the calculator in my screen and I'll do it.
sure, ma'am.
Can you see my calculator
No, ma'am.
now?
Yes, ma'am.
Yeah. So you have 1.4 divided by
27.
27, right? So this is coming like 0.05. You can write it as 0.05. So 0. Now what you have 0.05 into 10 raised to the power of minus 16 whole to the power of 1 by 4. Okay.
Yes, ma'am.
So 0.05 to the power of 1 by 4 into 10 raised to the power of minus 16 to the power of 1 by 4. Okay.
Yes.
So 0.05 to the power of 1 by 4. So either you write like this 0.0, sorry, sorry. So you have to take 0.05 to the power of 1 by 4. So 0.05 to the power of, you put brackets and then write 1 by 4 inside the bracket, okay, and press equal to. So you will get this answer 0.47. Okay, this is clear.
Okay. Yes, ma'am.
Or you can write it 0.05 y root of, and value of y is 4. Then also you will get the same answer 0.47. Okay, now 0.47 into 10 raised to the power of minus 16 to the power of 1 by 4. So 10 raised to the power of minus 16 to the power of 1 by 4 will be equal to 10 raised to the power of minus 4. Okay, so that means your answer is 0.47 into 10 raised to the power of minus 4 or 4.7 into 10 raised to the power of minus 5. What's the answer given?
Ma'am, answer is 5.2 into 10 ki 10 raised to the power of minus
Okay,
6.
then the answer might be wrong.
Okay.
Any other question?
No, ma'am, that's it.
Ria, you had any question from assignment 1, right?
Yes, ma'am. Ma'am, assignment 1, question number 12 and question number 13.
Can you share your screen if it's on your screen? Hello? Yeah.
Ma'am, is it visible now?
Yes, it is. So, question number
Ma'am, 12.
12. A 0.2 molar solution of a weak monoprotic organic acid is 2.3% ionized at 25 degrees Celsius. Ka value has to be calculated. Yeah. What doubt you have?
I'm not able to understand this question. How to do that?
Okay, so it's a monoprotic acid, right? So monoprotic acid means it has one proton. So it can be written in the form of Ha. That is fine.
Okay, ma'am.
And now that monoprotic acid is getting dissociated, so Ha will dissociate into H plus and A minus. So monoprotic acid means like acid like HCl. Okay, HCl will dissociate into H plus and Cl minus, right?
Yes, ma'am.
So similarly, any random acid Ha can get dissociated into H plus and A minus. Okay.
Okay, ma'am.
So now they are saying that they have started with a 0.2 molar solution and it's a weak organic acid, so it's not dissociating completely immediately. Okay, it will slowly dissociate. So they are starting with 0.2 molar. So at the beginning the concentration of the acid is 0.2 molar and H plus and A minus will be 0 because there is no dissociation immediately happening. Okay, and after
Okay.
the dissociation now they are saying 2.3% is ionized. Okay, so let's say some X value is ionized. So if two, like this 2.3%, you can just imagine it as X. Okay, or alpha. Here alpha is given, so we'll take alpha. So alpha amount is ionized. So that means alpha amount is forming. So we started with 0.2. Let's say we started with 100. Okay, so if I start with 100 and if I tell you that 2% is dissociated from it, that means what is remaining from 100? 98 is remaining,
Yes, ma'am.
right? And what is forming? 2% is forming,
2%.
right? So that means 2 is forming, 2% of 100 is forming basically, right? Similarly, if alpha percentage is dissociating, that means 0. Alpha of 0.2 will be forming. Did you
Yes,
understand?
ma'am. Yes, ma'am.
Okay, so 0.2 alpha is forming, and since it's a monoprotic acid, so same amount of H plus and A minus will form. So 0.2 alpha will be the concentration of both H plus and A minus.
Yes, ma'am.
And what is remaining? So remaining is basically 0.2 minus 0.2 alpha. So 0.2 alpha is forming out of it. So 0.2 minus 0.2 alpha. So if you take 0.2 common, so it will be 0.2 in the brackets 1 minus alpha.
Okay, ma'am.
Okay, now you have the value of alpha given as 2.3%. That means 2.3 divided by 100.
Okay. Ma'am,
Okay,
how come 2.3?
it's given in the question at 2.3%. Read the question.
Okay, ma'am.
So the value of alpha is given. Okay. So the value of alpha is 2.3%. So 2.3% means 2.3 divided by 100. That means 0.023. Okay.
Okay, okay, okay.
And now you have to calculate the dissociation constant, right? Ka. So Ka is equal to the concentration of the product divided by the concentration of the uh, reactant. So that means 0.2 alpha into 0.2 alpha divided by 0.21 minus alpha. This you understood?
No, ma'am.
What, how do we write the value of k? If you see in the question below that also, that is given in all the questions, you will see the k is solved as that only, right? How much is dissociated divided by how much is remaining. So what is the final concentration of the dissociated compounds? That is H plus and A minus. So final concentration of H plus is 0.2 alpha multiplied by final concentration of A minus, that is 0.2 alpha, divided by the final concentration of the initial compound, that is 0.21 minus alpha. So concentration of H plus into concentration of A minus divided by concentration of HA. This is clear?
Yes, ma'am.
Now this is clear how K is solved.
Yes, ma'am.
So now you have the value of alpha, so you can directly either replace the value of alpha. Now what they have said is, since the value of alpha is very very less than 1, so 1 minus 0.023 will be almost equal to 1 only, right? You can either solve this also,
Hmm.
but when you will solve it, you will see that 1 minus 0.023 alpha, 0.2, sorry, 1 minus 0.023 will be almost equal to 1. Do you agree?
Yes, ma'am. Yes,
Use a
ma'am.
calculator and do it. How much you are getting?
Ma'am, this only 1.06 into 10 to the power minus 4.
Okay. So you, okay, so you got the answer.
Yes, ma'am.
Any other doubt?
No, ma'am.
Okay. Remika, do you have any doubt?
No, ma'am. Thank you, ma'am.
Okay. So today was the last session. So all the best for your exams.
Thank you so much, ma'am.
Yeah. So we'll end the session now.
Ma'am, there will be more doubt solving section, or this was the last one?
This is the last one. Okay. That's why I'm telling you, if you have any doubt, you can ask now only. We can extend this session for 10-15 minutes more if you have any doubt.
Ma'am, assignment-based questions little come in the exam or not?
The question should be the assignment based only. I am not involved in that part, so I won't be able
Ma'am,
to
is there
answer
will
that.
be
But
negative marking is also there?
Again, I don't know the pattern. I think exam format they gave, right? So
Previously, there was no negative marking, but in Google, they are showing that in 2023 there is a negative marking.
okay, they have questions will be on computer. You will have to some. They haven't mentioned, but I won't be able to answer that question. The NPTEL team will be able to answer that question. Actually, we are just the TAs for this course, teaching assistants. So we just deal with the problem solving part of it. The format of the question paper you can mail the NPTEL team directly and ask me. Okay. Anything else?
No, ma'am. Thank you so much, ma'am.
Okay. So thank you all of you.
I'm having doubt in radioactivity, age determination portion.
Okay. What is the doubt?
Ma'am, that ma'am, concept I know, but I'm getting confused how to solve the questions. Like, I'm not able to understand the questions, but I know the concept of first law and second one.
Like you understand the half life, like how they are
Yes,
calculating
ma'am.
the half.
Yes, ma'am.
Yeah, so
I know that,
what is
but
the doubt you are getting?
I'm I'm getting confused in the questions what they are going to about to say like that.
So you have seen the assignment questions that I have discussed and the assignment questions that you guys have gotten this year,
Ma'am, in
right?
numerical
Yes,
part, I'm
numeric.
getting
It's
confused.
basically formula based. So if you remember the formula, you won't face a lot of difficulty. So mainly the formula where you calculate the half life from the, like, either the k value is given and you have to calculate the half life, or the half life is given and you have to calculate the k value. The second thing is that c0 upon c, that part you should, like, how much time it is taking to uh, decay
Ma'am, you
from the
find out the age.
original amount. Yeah, so finding out age also, it's formula based, right? You just have to see the formula and calculate. So all the questions are formula based. So I have already uploaded the slides on the drive. I think you guys can access it.
Okay, ma'am.
There is you know uh, the link is given to all the videos and the slides, right, on the portal, on the NPTEL portal.
Yes, ma'am. Ma'am, that problem solving sessions one.
Yes, yes, so you can go through my slides. So in the slides only I have mentioned the formulas for each rate reactions, and how to calculate the, like, if you see the numericals there, you will understand which all, like, which formulas you have to remember, and just remember those many formula and you will be able to solve the questions. If you
Ma'am,
have any, yeah,
ma'am, where we go to find your slides? Like, I'm like getting.
on the portal only, on the NPTEL portal. At the end where you have week 1, week 2, week 3, like that written on the left hand side panel, at the end you will see problem solving session written on it.
Solution of assignment, ma'am, we have, and
At
like,
the end, below that, below all the weeks tab, you will see problem solving session.
okay, yes, ma'am, I got it.
So in the
No
problem
video,
solving
it's
session,
possible. Can we see like if something can I see your video again if it's possible?
yes,
Like
yes, the videos are uploaded on YouTube and the links are given. So in the problem solving session, you will find a link to a spreadsheet,
okay.
right? So videos are given. There was some technical issue in the initial videos, so I have re-recorded those videos so you can see like most the questions are all the questions are discussed. There's some lag between the audio and the video, so you can just try to understand that, and rest you can see the slides also, so you can access all the sessions through the videos and the slides that I've taken.
How many questions will be there?
Again, I won't be able to answer that also. So these are the, if you want to know the exam pattern, I think you should ask the NPTEL team to send or check on internet if the previous year pattern is given. I
They are
think
saying
previous
like
year pattern will be
our
given.
teachers, our university teachers are saying that no negative marking will be there, uh, because they have asked to some experienced NPTEL members that uh,
Then
and
it won't
in
be there. If
Google.
they are saying I'm like, I think that it won't be there. They must be knowing someone from the NPTEL team. That's why they are telling.
But in Google, they are saying that from 2023 there will be a negative marking. But I have to sure that how much, like, what is that marking like of negative?
I
How many mark
also
will be
don't
deducted?
have the idea for that, but if let and like I really can't say anything. If you can mail the NPTEL team, if they can reply, that's fine.
Okay, ma'am.
Okay.
Ma'am, where we'll get the mail?
There is a mail ID given on the NPTEL portal only. I think there must be a mail ID saying like for help purposes. Let me
Okay,
check.
ma'am.
Just check on the portal. You will find. Okay.
Okay, ma'am.
Yeah. So prepare well for your exams. From which universities are you guys from?
Ma'am, I'm from Sharda University, Greater Noida.
Okay. And Ramika?
I'm from Vellore
Okay,
University in
VIT.
Chennai.
Okay,
Yes.
great. Okay, then prepare well for your exams, and
Sure,
for
ma'am.
the exam pattern, I would suggest mail NPTEL team once. They will clarify, and or just ask your professors if they know anyone. The IIT Madras is mainly conducting, like managing this. So if they know anyone from IIT Madras, they can directly contact and ask.
Ma'am, don't know anyone from IIT Madras.
No, you won't know. Your professors might know. That's what I'm saying.
Okay, ma'am.
Yeah.